I will also talk about biostimulation using DOC, or dissolved organic matter, as Rita mentioned. But in this case, I will also talk about biostimulation usage as a tool to assess what is actually the biodegradation capacity of microorganisms in the field. Yeah. So very briefly, because you already had a very nice introduction about what is the current situation in the Netherlands with the presence of pesticides, I just want to maybe strengthen the fact that actually a lot of the groundwater present in the country is used for drinking water production, and unfortunately pesticides presence is threatening the groundwater quality and therefore drinking water production. So of course we as scientists want to come up with a solution to address this problem, and I think one of the solutions is bioremediation. So bioremediation relies on biodegradation natural processes, and of course for biodegradation to occur we need a balance between the contaminant concentration or availability, which is something that was also discussed before, the environmental conditions, and of course ideally when you have contaminant availability but also good environmental conditions, then you can develop a biodegrading microbial community that is active. And as Rita discussed before, then one of the challenges is, of course, that the groundwater environment is a challenging environment for microbial communities to develop. But next to that, also the contaminant concentration is it's very low. As it was mentioned before, it's in the micrograms per liter concentration. And of course, that's too poor for microorganisms to rely fully their growth on pesticides. And I think I want to also mention something about the redox conditions. So these are the environmental conditions that are present normally in the aquifer. So this is really textbook, what we would expect in the top: nitrate reducing conditions, iron reducing conditions, and as long as you go deep in the aquifer, you will also find sulfate reducing and methanogenic conditions. And in a previous field study that it was conducted also from our group, we found that different redox conditions shape the microbial communities. So as I was mentioning before, then we need to try to achieve this balance. And then we already found that different redox conditions in the field are the main selective pressure that microorganisms in the field have. And that is why we wanted to study what is actually the biodegradation capacity of the microorganisms in the field. And this means, like, if we have the naturally occurring indigenous microbial communities, then what is their capacity to actually biodegrade pesticides? So we know that they've been in contact with these pesticides, and we wanted to figure out how does this pesticide influence their microbial activity. And for this, we did a batch experiment as well. In this occasion, we tested 4 different amendments because, as mentioned before, the groundwater conditions are very poor. And what we wanted to do is to improve a bit environmental conditions to give the microbial communities a boost for their activity. So we tested nitrate addition and DOC or DOM, what Rita presented before. We also tested with oxygen and DOC addition with oxygen and only with dissolved organic carbon. And for conducting this experiment, we went to monitoring wells. So these monitoring wells are indeed around extraction location. And basically, we selected 2 wells that are relatively close to each other. They are in the northeast of the Netherlands. It's an agricultural area. So again, of course, we expect pesticides to be there. And we sample at 5 different discrete depths, so at 5 different levels from around 13 to 65 meters deep. So basically we wanted to sample all the redox conditions that were present in the field. And this is an explanation of how we conducted these experiments. So again, we have the 4 amendments, so nitrate and DOC, oxygen and DOC, oxygen and DOC alone. We also have a blank. Blank, it's basically only groundwater without any amendment. And we had some sort of groundwater control. So basically we had millicube water, and then we tested the amendments of oxygen plus DOC and nitrate plus DOC. And the pesticides that were tested were basically BAM, 2,4-D, and MCPP, or Mecoprop. And the concentrations of DOC and nitrate that we used in our experiment are similar to field concentrations. And this is a summary of my results. So basically, from the groundwater samples that we took, with the different amendments, we only observed removal of 2,4-D, and this happened only when oxygen plus DOC was added to the batch bottles. So this is the general trend. We didn't observe any degradation only with oxygen or only with DOC, but mainly in the combination of oxygen and DOC is when we observed biodegradation occurring, and therefore we also think that it is important for microbial communities a good electron acceptor, so in this case, of course, oxygen, but also a carbon source that they can rely on instead of the pesticides that, as mentioned before, are in really low concentrations. Something else that we observed and is quite interesting, it's basically in these figures, you observe in the upper part, it's only from well 22, in the lower part is from well 23. And the 22.1 will be the most shallow deep, and 22.5 will be the deepest section. And basically what we observed in the oxygen plus DOC amendment is that the 2,4-D degradation changes depending on the depth. So this shows as well that the microbial community activity also depends on where are the microbes located in the aquifer. So something similar to what Rita mentioned before, the fact that they are in a more shallow section, probably they are more used to be in contact with carbon, but also with pesticides, and therefore they are a bit more active. So for example, we can observe that 22.2, so the second depth, which is around 23 meters, is the one that has like a faster 2,4-D degradation compared to the other depths. And then we also observed that in the well 23. So I think indeed we can conclude that the degradation capacity is definitely depending on the environmental conditions that microbial communities have in the field. And this brings me to my take-home messages. So first, I want to strengthen that the groundwater is an unexplored environment, so there is many research still to be conducted. But we found degradation potential. Of course, also one of my take-home messages is that, unfortunately, the environmental conditions that are in the field are not really enough to keep up with the good microbial activity, and therefore they need to be improved so that pesticides can be biodegraded. And finally, that biostimulation has potential for field application. And with this, I would like to also acknowledge the co-authors, but also people that helped me with sampling in the field, which was also a very nice experience. And yeah, I want to thank you all for your attention, and I'm happy to answer some questions. Thank you.
Thank you, Andrea. I see in the pictures that the field sample was also in the autumn. Was it autumn 1 year ago or 2 years ago?
2 years ago,
2 years
yes.
ago. Okay. Are there questions from the audience to Andrea? Yes.
I can still talk aloud.
Yeah?
Did you probably also look for microbial community composition, like what organisms you find in these?
Yeah. So not exactly for this experiment, but as part of my PhD research, then also we did some sequencing with the same wells. And that's also why one of my take-home messages is that groundwater is a very unexplored environment. So as a summary, we found out a lot of unclassified sequences and therefore unknown microbial communities, non-known microorganisms. So I think that's definitely one of the things that need to be further studied in the future. So we are now a bit limited because of the lack of information in databases, but I think as more people conduct research, then we will have more sequences available and probably a better classification of our microbial communities.
Hello. I came in a bit later, so maybe you explained it already at the beginning. But for the deeper depths, did you also consider using sulfate as an electron acceptor for the lab tests?
Yeah, so we know from also monitoring information that sulfate was present. So basically we added nitrate additional to the naturally present sulfate that was there. And in some other part of my research, then I also had a sulfate column and a nitrate reducing column to emulate better the field conditions. But we still didn't find as promising results with sulfate so far. Yeah.
A question from my side. You showed that depending on the depth, the degradation was quicker, at least at this second point, but not at this first point. And in this first point, you would expect the highest concentration. So do you have an explanation there?
Yeah, well, what I think is also that probably pesticides travel very fast to the second depth because of rainfall and all these like natural phenomena. And that's why I think, well, we actually presume that in the upper part of the aquifer we have vertical flow. So that means that the transportation of pesticides will be very fast in the upper section, and we hypothesize that there is actually horizontal flow in the deeper section. So yeah, that would be one of the reasons.
Did you measure the actual pesticide concentration at the various depths?
Yes, so we have like historical monitoring data from a drinking water company, but the separation between shallow and deep is more based on the soil profile, but also on the different microbial communities we found. Yeah.
Are there still other questions by the audience? If not, then I want to thank you too.
Yeah.